Hi, Jamie. Can you hold up the room, please? Because
Sure.
the sharing link doesn't work again. Is somebody maybe on the desktop version? No. Okay, I'll leave and come back in and try to restart the app. Okay.
Do you want me to stay here? Yeah?
We're all
Yeah.
here.
Hi, Victoria. Hi, Serena.
Okay.
Everyone's
Hello.
leaving.
Hi, Victoria.
Hey,
Hi,
Jamie.
Serena.
Hey, Serena.
Hi, Victoria.
Hello.
Hey, Jamie.
Dr. Balder?
Hello, Dr. Balderston. You're on mute. To unmute, it's in the lower right corner.
How about that?
Great.
There we
Hello.
go. Hiya. Hi, Doctor. Pleasure to meet you.
Thank you all for having me. This is going to be fun.
We're so glad to actually have you here today. Have you had a good day so far?
Yeah, it's been good. I have a grant that's undergoing council review right now, or what was earlier today. So I keep checking the website, but nothing. So it's a good day, but
Hi.
yeah, I'm waiting.
Well,
Oh,
best of luck with that, yeah. And welcome back, Katarina.
Thanks.
waiting for the grant
Okay.
review committees. Yes, we shall distract you.
I appreciate that.
Serena?
Beautiful.
Oh, she's just fixing some bugs with her app. She'll be here
Shall we?
momentarily.
Oh, sorry, Jamie. Go ahead.
Oh, no, please, Victoria.
I was
Please
just
go
going to say,
ahead.
should we do a little stage dive and then bring Katarina back up into upper left? Opinion, Katarina?
Hi. Sorry. I was now my app works. I had to update it because I couldn't pin the presentation.
It looks
How are you? Welcome, Nicholas. Thank you for coming.
Yeah, thanks.
I'm sorry, I opened the room, but then I had to drop out and come back in. I wasn't able to post to put the presentation up, so I wanted that to work for sure. So thank you for your patience and thanks for being
Yeah,
here.
no problem. It's funny. I've been back in the office for a while now. So when I came upstairs to get ready, I had to clear all the little toy trucks and little figurines and stuff off of the work from home desk, which now doubles as the Paw Patrol desk before we got started. So I'm glad I gave myself some extra time.
Oh yeah, it got it clogs up everything fast with kids. They take over
Yeah.
every little corner. Yeah.
Are your kids going to be listening to talk this evening?
If we're lucky, they'll stay asleep. Well, one's asleep. He's been asleep since like 5. He had a busy day at preschool, and I think the other one, she had a nap, so she's downstairs with mom. She'll probably stay down there. She'll probably go to sleep at some point. Hopefully I don't disturb them. But
Okay, we'll keep our voices lowered.
no,
That's a
it's
joke.
okay. If they come in, they come in.
So I've been hearing about TMS for a while. I'm going to be really curious what the latest word is.
I think it's really exciting. It's a really really exciting, like, new emerging field. At the same time, like, it's it's been approved for depression for going on like 15 years now, or pretty close to that, I think. So it's showing like some clinical efficacy, but there's still so much to explore. So it's a really cool field. I'm excited to be a part of it, and be able to have the room to play with the parameters a little bit and explore a little bit. I think that's a fun position to be in when you're doing science.
Oh yeah, I will probably get around to asking you about the interaction with calcium waves and astrocytes at some point. But We can easily do
I
that.
all right.
That's Serena's way to warn you what's to come, so you can be prepared for it, because there's going to be a lot of
Well,
questions.
I slipped and fell
Okay.
in the astrocyte rabbit hole, so I'm just really fascinated with the whole bit. So there's usually an angle like I'll when I find a good setup.
Well, I'll have to. All right. Don't mind me googling in the background because I don't think I know the answer to that.
Oh, okay. Well, yeah,
But
that'll be even more fun.
The guest,
I,
Serena. Tonight's episode of Stump the Guest.
Well, no, it's astrocyte syncytium, calcium waves. They're connected with gap junctions. Anyway, yeah, okay.
Now this will be.
Hi, Kirko. Hi, everyone in the audience. Hi, Summer. Hi, Jenny, Joanne, Simon. Welcome, everyone. Please feel free to ask or comment questions in the chat or come up to the stage later if you have questions. And we will start in a minute. And so, yeah, thank you, everyone, for coming. And of course, Nicholas has a special thing here. Oh, yeah, it's already 9. I just realized. So, yeah, I think, can we get started? I think so, right? Cool. So welcome everyone to the Science Society. It's another wonderful evening, and we have an amazing guest speaker here today, Dr. Nicholas Balderston. He will talk about his novel individualized targeting in TMS treatment. But before we start with questions and the presentation, let me introduce you to him. He did his he's a research assistant professor of psychiatry at UPenn, and he did his bachelor at the University of Florida and his master's at the University of Wisconsin, and later his PhD in experimental psychiatry and neuroscience at the University of Wisconsin. And later he did his postgraduate training at as a Chateaubriand Fellow at the École Normale Supérieure, and his postdoctoral fellow as a postdoctoral fellow at the NIMH from 2014 to 2019. And Dr. Balderston is an experimental psychologist, and his work mostly focuses on anxiety, and his research uses different approaches for analysis, like psychology, neuroimaging, neuromodulation, to test brain behavior hypothesis, to understand the mechanism that lead or to clinical anxiety disorders. Yeah, so thank you so much for being here and talking about your really important work. But before we get started with your presentation, Victoria would like to ask you a few more general questions, if that's okay.
that's wonderful.
All right, thank you, Katerina, for the bio. That was so fascinating. And so, yeah, Dr. Balderston, to carry us into your discussion tonight, I just have a few questions for you that are to learn a little bit about you and to help us get to know you a bit. And so my first question is, if you can reflect on your life and this any time in your life, and out if a moment or an experience that really let you know, that clued you into the idea that you felt connected. Ryan, that is my question.
Okay, so I think on mine it cut out a little bit, but if I understand you, you're asking to point to a time in my life where I felt connected to science. Is that the gist of it?
Yes, you out a little bit. I don't know if it's me. Let me go my what. Yeah, that was my question.
Okay, so I think I didn't have a background in science, and really until I got to grad school, I didn't really know that that's something that you could do with your life. And I think the first time I really felt that was in the final semester of my undergrad. I was taking a class on brain behavior relationships. It was essentially an undergrad intro to neuroscience class, although it wasn't really labeled as such. And the author of the textbook wrote it in such a way where they were talking about different discoveries made by different people in different labs and sort of the conditions of the lab and how the research got done. And they really laid it out as a job that people did in real life and not this sort of you know fantasy thing that happens in the movies by people in like white coats. And until then I'd never really thought that that was something that you could do. And so I was just really really fascinated with that book, and it made me want to get into neuroscience, and it made me want to study neuroscience. And it took me a couple of excuse me, took me a couple of years to get to the point where I was able to make it into grad school and able to join a research lab. But when I did, it was great. It was so cool. I showed up. I showed up in Milwaukee on a Thursday, and I called my soon-to-be advisor, and I was like, "Hey, I'm in town. Maybe we could have a meeting or something, and I could get to know the lab." And those are the first words that he spoke to me. He was like, "Well, tomorrow's Friday. We have lab meeting. It's at noon. You're expected to be there." And so I showed up at lab meeting, and from then on I was like in a lab and I was doing science. So I don't know. It's kind of a long-winded answer, but that's sort of how I got my start, I guess.
That's the best kind of answer. Thank you. We
Sure.
appreciate that detail because all the details, they're like the beads in the necklace,
Yeah.
that make it add, glimmers of and sparks of interest. And I think it's interesting to hear how many times we think that something that's really just fun for us isn't something that's accessible. And that sounds like what you were saying, that this book showed you that science was a real thing that you could really do. And what was the name of the book?
Oh, I have no idea at this point.
Okay.
I think it was. I think the one of the authors was I forget his first name. Koob. I think he's I want to say that he's in the intramural program now. I think he's one of the maybe he's the leader of NIDA, or not NIDA, maybe in INDS. I don't remember. I think we're the authors of the textbook, but it's been since probably 2004 since I read that.
Well, thank goodness you came upon that. And then maybe you can bring us along the path from that Friday lab meeting up to where you are in your current research.
Sure. Yeah. So yeah, I showed up at lab meeting on Friday. He introduced me to Doug and Jennifer, and they were the senior grad students that were going to teach me. And so they showed me to my office, and I had an office, and I had a computer, and I had access to fMRI data and neuroimaging data. The lab I was in was a classical conditioning, like fear conditioning lab that did sort of both human and non-human animal research, and the human research was all fMRI of fear conditioning. And so I started out by analyzing some of the data sets that were collected and started programming some of my own psychophysiology experiments. So I started out doing, fear conditioning and measuring skin conductance responses from the fingers. And then, did some fMRI work. Through happenstance, I met the then head of the MEG, so magnetoencephalography research program at MCW, and realized that was a cool new technique that I could use to study really rapid responses. And I wanted to study really rapid responses in fear learning without awareness. So we started doing a collaboration there, and then he introduced me to my one of my other pre-doctoral mentors, Dr. Catherine Talon-Bowdry, who's at ENS at École Normale, and so I did a fellowship there, and I collected all my dissertation data using MEG in Paris, and I lived in Paris for like 9 months, which was really fantastic. If you ever get a chance to somehow find a grant that takes you to Paris, like, yeah, I highly recommend it. And then I came back and I defended my dissertation, stayed another semester in Milwaukee, and just started applying for postdoctoral positions. And one of the places I applied, and one of the people I reached out to, was Christian Grion, who led a section in the NIMH intramural program. And he was looking for a postdoc. His work was mostly using the threat of predictable and unpredictable shock. So it's really similar to fear conditioning, but it's more instructed, and it gives you a way to sort of differentiate between really rapid fear responses and more like phasic, sustained anxiety responses. But what was really exciting for me is it gave me a chance to combine the psychophysiology type experiments with more cognitive experiments. So one thing you can do with that is you can layer, threat processing on top of other cognitive tasks like working memory tasks or executive processing tasks, that sort of thing, and really study the interaction between anxiety and cognitive processes. So I did a bunch of studies in Christian's lab. I'll also say one of the really nice things about working in the intramural program is the resources are pre-allocated. You don't have to apply for grants or anything. You're just given time on the scanner, and you're given a budget, and you can really do a lot of really interesting research that you would otherwise have to, scrounge and figure out a way to pay for. So, as a postdoc, I was a postdoc there for 5.5 years, and I think I probably collected, several hundred subjects worth of fMRI data, just because we had the time, and that's been so valuable as like an early career researcher now, to have that data as a windfall to sort of look back at and use as like preliminary data and really design new studies. But while I was there, we did quite a few studies looking at anxiety cognition interactions, and we kept finding this one region of the brain, the dorsolateral prefrontal cortex, that was becoming activated when these two things sort of, let's say, butt heads. And so what we wanted to do is we wanted to stimulate that region and see what happened. And so we formed a collaboration with Dr. Holly Lisenby, and I was able to start learning neuromodulation and brain stimulation and TMS and all of these really advanced methods that I know now. I just got some really, really excellent training, and we were able to find some interesting interactions, or interesting causal interactions, where when you perturb that circuit or when you perturb that region, you can directly impact anxiety expression in a systematic way, which was really interesting. And so, collaborated with her, worked in her lab for a little bit, and then she introduced me to the head of the center that I work at now, and I was able to find a soft money position in the psychiatry department at UPenn, through that connection. And now I'm just working on finishing up a career development grant, and I'm looking to transition out of that career development phase and start applying for research grants from NIH. And really, the next 5 years of my life are just going to be to secure enough of those grants to start building the lab and start building a research program, and we'll see how it goes. That's kind of it.
Yeah, thank you. Thank you for bringing us along. And I hope, and I think every one of us has noted your advice about making sure to find a grant that will send you to Paris. So thank you. That's great advice. Beyond that, you're welcome to begin your discussion, and we can follow that with Q&A, if that's okay with you. And you can relax, and we have your link pinned at the top, and we will take care of modulating questions and that. So thank you so much, and the mic is yours.
Thank you.
Thank you.
Okay, that's great.
Yeah, you're free to begin.
I'll just really jump right in then. Hang on just a second. I'm gonna shut this door. As you might have guessed, today I'm going to talk about neuromodulation, and I'm going to talk about how we can use data that we know about the brain, specifically functional connectivity data, to try to understand the best way to target and deliver neuromodulation therapy to precisely impact symptoms in individuals and groups. And if we jump to the next slide, to slide 2, my background, as I talked about before, is in anxiety. But anxiety and depression are really comorbid, and together they make up the largest category of psychiatric disorders in terms of total number of diagnoses. And depression is also one of the largest disorders that lead to significant impairment. So 5% of the population suffers from depression, but it's the leading cause of disability worldwide. Leads to suicide, or can lead to suicide. Over 700,000 people die of suicide each year. And anxiety is also really impactful. I think the overall impact of anxiety on society is upwards in the $48 billion per year range. And we have treatments for both depression and anxiety. The primary treatments are medication and talk therapy, so like cognitive behavioral therapy or exposure therapy. But there are also barriers to treatment. So a lot of the drugs that we use now have side effects. You have to try one drug and then the next drug and then the next drug, and really, it's trial and error to figure out which medications work for folks. And therapy can be expensive. It can be hard to make all the appointments, and we can do better. We can try new treatments, or we can try augmentation treatments to try to improve these existing therapies. And that's where neuromodulation comes in. And when I talk about neuromodulation, let's jump to slide 4 now. Really what I'm talking about is device-based neuromodulation. And so the idea is that you're going to use some kind of energy, whether it's like electric energy or magnetic energy or pressure energy in the case of, or sound energy in the case of focused ultrasound, to impact specific circuits in the brain in specific locations while leaving, other locations and circuits unaffected. So unlike pharmacological interventions where you take a pill and that sort of is processed by the liver, gets into the bloodstream, and really affects to some degree the entire brain in a similar fashion. These device-based neuromodulation techniques are more focal, and they don't involve any chemicals being entered into the bloodstream. So in terms of side effects, the side effects are pretty mild. If you go to slide 5 now, you can see really this is sort of the state of the art in terms of what's being done with neuromodulation. And you can see, if you look on the y-axis, you can see sort of the focality of the particular technique. And then on the x-axis, you can see the invasiveness of the technique. So, you can start out with something like transcranial electric stimulation, so like TDCS or TACS. These are what we call subthreshold, meaning that they can't really trigger any action potentials in the cells. So they're really just modulating the background excitability of the cells in the brain, but not really triggering brain activity per se. And with these electrical techniques, they're going to be much more non-focal because they're filtered through the scalp and the skull, and there's resistance there. So if you move to the right a little bit, we have what's called like subconvulsive. So these are techniques that can actually change the activity of the brain, so they can induce action potentials. But they're not really designed to induce a seizure per se. So again, they're still very non-invasive. Side effects are minimal. But in the case of TMS and DTMS, which is deep TMS, they're using magnetic fields, which can penetrate through the scalp and the skull much easier than electric stimulation. So you can get more focal activation of specific brain circuits. And then I won't talk too much about these other categories, but you have convulsive therapies like ECT, which has been the gold standard for depression treatments, for close to 100 years. So in the instance where nothing else works, ECT is a really profound and powerful way to make depressed people better. And we're really just starting to use magnetic energy in a similar way to induce a seizure to treat depression. This is known as magnetic seizure therapy. Again, the nice thing about magnetic seizure therapy is that you're not dealing with all the resistance of the scalp and the skull, and so you can get really more focal stimulation and potentially minimize the cognitive side effects of ECT. And then finally, we have deep brain stimulation and vagal nerve stimulation. These are going to be the most invasive, because you actually have to do surgery to implant these devices. So this is just sort of a background of the state of neuromodulation as it exists today. My talk is going to be focused on TMS, and that's the technique that we use in our center primarily, and that's the technique that I use in my lab. So if you skip to slide 6, you can see myself and my very enthusiastic, we'll call him an unpaid intern, who is administering the TMS. So the way that it works, you can see there's a coil, a figure 8 coil, and you place the coil near the scalp, and you activate the coil, and that activates the brain cells directly beneath the coil. And different types and patterns of stimulation could lead to different changes in activity. So there are some types of stimulation that can decrease the excitability of a particular region of the brain, and other types that can increase the cortical excitability. So I like to think of it as, in the case of excitatory stimulation, it's almost like you are training a particular region of the brain. So you are sort of giving it exercise to build up the connections in a similar way as you would do if you were like lifting weights or something like that. The principle of TMS is based on Maxwell's equations. I'm not really going to go into these, but there are a couple of fundamental principles. One is that particles and fields are essentially interchangeable, as are magnetic fields and electric fields. But importantly, they interact with one another. So if you look down at Faraday's law, the main takeaway is that when you change a magnetic field, you induce an electric current. And then with Ampe's law, changes in currents or electric fields can create corresponding magnetic fields, and that's how TMS works in principle. So that's slide 7. If you jump to slide 8 now, you can see it in practice. Again, this is a standard administration of TMS with the coil near the scalp. If you jump to slide 9 now, you can see the electric current is being passed through the circular coils that make up the figure 8 coil configuration. And in slide 9, you can see through Ampe's law, this creates a magnetic field that is perpendicular to the coils that penetrates into the brain. And it penetrates directly through the skull without any resistance, again, because it's a magnetic field and not an electric field. And then if you go to slide 10, if you go to slide 11 now, you can see with Faraday's law, once the magnetic field penetrates into the brain, it also creates a corresponding electric field that affects the excitability of the neurons directly below where you place the coil. And so knowing these fundamental physical laws, we can then try to understand where in the brain the electric field is going to be created, so that we can best position the TMS coil to target the specific brain regions that we're interested in. And the way that we do that is shown in slide 12. The way that we do that is we essentially solve these field equations for this complex system. And so the main method that people use for doing this is called finite element modeling. The idea is that you take this complex system, which is the brain, the head, the skull, the scalp, the CSF, the gray matter, the white matter, and you tessellate those by sort of wrapping a triangular tessellated mesh around them. And then for each of the points on all the triangles on those meshes, you solve Maxwell's field equations. And if you do that, you can see the distribution of the electric field in the brain. And I'm hoping you can see in slide 13 the animation. You can see down in the left-hand corner the position of the figure 8 coil, just represented as a bunch of single point dipoles. And then in the larger picture, you can see as the coil rotates around and around and around, both the direction of the current and also sort of the intensity of the current in the underlying cortex changes in intensity. And what we can do then is we can use this electric field distribution to understand how TMS might change the underlying activity of the brain. And then we can solve these equations for multiple locations and multiple orientations of the coil, and then evaluate those in some way to figure out the best way to hold the coil to directly impact the part of the brain that we're most interested in. And those two key points are what I'm going to focus on for the rest of the talk. The first one is, figuring out how the electric field is going to penetrate into the brain and distribute across the brain, and then figuring out, the best part of the brain to try to stimulate. So those are the two key fundamental problems that I'm trying to solve with my research. But if we jump to slide 14, we're gonna and then to slide 15, we can start to look at applications of TMS, and even even in the early days, before we started doing these more fancy electric field modeling type approaches to optimize the TMS coil positioning, you can get a pretty good effect just by placing the coil where you think is a good spot. If you know that the dorsolateral prefrontal cortex is important for depression, if you place it over the DLPFC, chances are you're gonna impact depression. And so that's kind of how the early studies were conducted, using what's known as like the 5 centimeter approach, where you find out where the motor cortex is, and then you just move the coil 5 centimeters across the scalp in anterior direction, and you aim for that spot. Again, this isn't the ideal approach because it's not really optimized across participants, but using this approach, researchers were able to show that it significantly impacts depression symptoms when compared to a sham TMS treatment. And the study that I'm showing now is the original study that led to the FDA approval for TMS as a treatment for depression. And this has been replicated numerous times, and it's been validated using meta-analytic approaches. And so we have a pretty good idea that stimulating the left dorsolateral prefrontal cortex can improve depression symptoms in treatment-resistant depressed individuals. However, again, I think we could do better. If we jump to slide 16 now, again, the major focus of my work is to try to optimize the targeting of the TMS coil, both in terms of the location, so the physical point in three-dimensional space, the XYZ coordinates. What's the best location to hold the coil? But then also the orientation. So if you think about it like a plane, you've got the roll, the pitch, and the yaw. And all of these 6 degrees of freedom need to be accounted for if you're going to optimize the coil position. And so the question then is, what are the data that we use to optimize these 6 degrees of freedom to best position the TMS coil for treating depression, both in individuals and in groups, or really any psychiatric disorder for that matter. So we jump to 17. The research that I'm going to talk about uses a resting state fMRI. So if you're unfamiliar, slide 18 is sort of just a brief primer in resting state fMRI. So the idea with resting state fMRI, the idea with fMRI in general is that you're recording changes in blood flow and blood oxygenation in the entire brain across time, and then you split the brain up into different chunks, different cubical chunks called voxels. And then you can record the signal in that voxel over time, which corresponds to the amount of oxygenated hemoglobin in the tissue there, which roughly corresponds to the activity level. But with resting state fMRI, the approach then is to take that signal that's recorded in that specific location in the brain across time and correlate it with the signal recorded from another location in the brain across time. And if you do that, say, in left and right motor cortex, what you can see, let's say in panel C with the yellow line and the orange line, is that across time, the signal in those two voxels are highly correlated. And, I think there's a lot of work going into trying to understand what that means. But what we do understand is that this correlated signal across time is to some degree related to both structural connections in the brain and also communication in the brain. And so, it serves as an okay stand-in for understanding how different parts of the brain communicate with one another. And it roughly corresponds to the types of activation you would see if you just had somebody do a task that activated those voxels. So tasks that activate, the left motor cortex are generally going to activate the right motor cortex as well. So the resting state data tends to also reflect the task-based data. So in this simple case scenario, we're just basically taking one voxel on the left side and one voxel on the right side, and we're correlating with those with one another. But we really have the whole brain, and the whole brain is split up into voxels, and we really want to know how all the voxels or all the locations are communicating with all the other voxels and all the other locations to see if we can pull out the maximum amount of information from these signals that are measured across time. And again, a lot of work has gone into, if we jump to slide 19 now, a lot of work has gone into understanding the best way to divide up the brain based on, coactivation studies, based on resting state functional connectivity studies, based on, data reduction techniques. And, broadly speaking, you can divide the brain and you can divide the cortex up into parcels. And using some of these advanced analytic approaches, you can group these parcels into networks. So really, I'm just showing in slide 19 one example of a brain parcellation and a brain network approach that divides the brain up into parcels that are grouped into networks that tend to correlate with one another across time in resting state fMRI studies. Now, the cool thing is, let's say that you wanted to understand how all of these regions are talking to one another, and you wanted to see if there's any sort of underlying information or structure, what you can do is you can correlate the time series in each parcel with the time series in every other parcel, and then group those parcels into networks and just display them as a cross correlation matrix. And so if you jump to slide 20, this is what I'm representing here. So this is just a standard functional connectivity matrix. And again, the value in this matrix represents the correlation coefficient between each parcel and every other parcel in the brain. And you can see I've just arbitrarily grouped these into the different networks corresponding to the labeling in the atlas that I chose to use for this. But when you do this cross correlation and grouping into different networks, you can see a very orderly structure. So you can see, across the diagonal, there are squares that are really bright, some that are dark blue. There's a lot of structural information buried in these functional connectivity matrices. And it's really the challenge then is, how do you best represent that information, and how do you best use that information to understand, the best spot in the brain to try to hold the coil and to try to deliver the TMS. So this is the start of answering that first part of the question. How do we use the information about the brain that we know to target the particular networks that are involved in the expression of, say, depression or anxiety symptoms? If we jump to slide 21 now, you can see that this functional connectivity approach has been applied to TMS targeting by other groups. And in the case of depression, what has been found is that if you target regions of the left dorsolateral prefrontal cortex that show a strong negative correlation with the region known as the subgenual anterior cingulate cortex, represented as this yellow dot in panel A, what you see is that people who have the stronger anti-correlation tend to have the best clinical improvement in terms of their depression symptoms. So if you place the coil in such a way where you're maximally targeting the negative correlation between these regions, the DLPFC and the subgenual anterior singular cortex, say represented by the green circle in panel B and the green dot in panel, or the green circle in panel B and the green dot in the green circle in panel B and the green dot in panel D, you could see that strong negative subgenual DLPFC connectivity is associated with good clinical improvement. Whereas if you target a different region that's positively correlated with the subgenual anterior singular cortex, you see much less clinical improvement noted by the red circle and the red dot in panels B and D. And this is shown in E just as a group level correlation. And again, what this suggests is that the underlying connectivity of the brain can tell us something about how depression is mediated and how to best target those networks in such a way to treat those symptoms. So if we go to panel 22, we're going to jump into my work now, and really the rest of this talk is I'm going to show you my approach to try to optimize those 6 locational degrees of freedom to target psychiatric symptoms. And I really like the work that was done in panel 21. It's really groundbreaking work, and it's been replicated a number of times, but it's specific to depression. So if you have some other psychiatric disorder, say like addiction or PTSD or anxiety, they may not share the same underlying mechanistic network dysfunctions. And so what we learn about depression may or may not be useful for understanding how to target for these other psychiatric disorders. So what I wanted to do is develop a method that you could generalize across disorders. So something that wasn't really dependent upon the type of symptoms or the type of network dysfunction. And so now I'm going to talk about this generalized approach. And really it begins in slide 23. So at its core, this is basically my approach. So if you look in the green panel here, the idea is that specific connections between regions of the brain are going to be important for understanding psychiatric symptoms. And you can represent how important a specific connection is for a given psychiatric symptoms by basically across subjects trying to predict the psychiatric symptoms based on the connectivity values for that connection. So in this case, the connectivity values for a given subject would be the X in this equation, and the slope of the line or the strength of the connection between symptoms and connectivity would be represented as the M, and then the intercept is B. So the idea is you can just use a simple regression equation to try to predict mood from individual functional connections. But then if you jump over to the blue panel, what you can then do is try to figure out how TMS is going to change those connections, and then modify the equation such that you introduce a new term into the equation. Let's call it E for simplicity for the E field, to then try to predict the change in mood and the change in symptoms based on how you think the TMS is going to affect that specific connection. And I just want to check in. Is everybody on board with this? Does this make sense? Is this confusing to people right now?
I'm okay, but does anyone have questions? Oh,
Yeah.
thank you.
So this.
Oh, Serena,
yeah, go ahead.
did you want to ask?
I just wanted to confirm it's making sense to me as well.
Hey, cool. So if everybody is really on board with this basic approach, now I'm going to jump in and go down the rabbit hole a little bit more. So again, the idea is you want to predict mood from connectivity values, and then you want to predict the change in mood based on how you think the TMS is going to affect those connectivity values. And then you want to scale it up to the whole brain. So, in just a basic theoretical approach, it should be as simple as, solving a big multiple regression now, where you include all of the connections in the brain. So you sum across, um, all the connections in the brain, and then you put that into a big multiple regression model, and then you predict mood from that. And then you downsample the. So then, if you go over to the blue panel now, you downsample the electric field model to the different parcels that you use to do the functional connectivity, and then you introduce an E term for each of the connections in the brain, and then again you sum across all of that to get the change in the mood. Ideally, it should be as simple as that. The downside to this approach is that you would need at least as many participants as you have connections, and in the parcellation that I'm using, that's about 5,500. So that's not really feasible. So we need to figure out a way to reduce this connectivity data in a way that's manageable. And I'll get to that in just a second. But first, if you jump to, sorry, that was slide 24. I didn't mention that. So if you jump to slide 25 now, this is really just how I'm coming up with the E for the equation. So you compute the electric field. You then have, so if you look at that, the blue brain to start out with, you essentially have a value for each voxel in the brain for how much field is going to be introduced into that voxel given your site of stimulation. You then downsample that to that same atlas that you use to do the functional connectivity that you use to parcellate the brain. And then essentially for each of those parcels, you have a value, and you can then convert that to a vector, which is the black and white line that you're seeing third panel over. And then you can essentially, let's say, average that vector with itself. So if you shift that vector 90 degrees and average it with itself, you can come up with a matrix that's sort of the same shape as that connectivity matrix that I showed you before, where each value in that matrix represents the average electric field um, in each of the two pairs of those connections. So the average across the pairwise comparisons. And really, if you boil it down to like the simplistic, as simple as it gets, the assumption that I'm making here is that the change in connectivity in these connections is going to be directly proportional to the average of the electric field in these two regions. This may or may not be true, but this is an assumption that I'm making with this model. So you can sort of see that represented in this graph here, where all the lines are just connecting each region to every other region. So this is how we get the E in the model. Now, to get the M and the X, we need to reduce the data. And so now we're going to jump to slide 26. And this is going to be an eyeful. So just strap in. So what I wanted to do is reduce the data in such a way that I capitalized on all that structural information that we know is in this large panel in panel A, but gets rid of a lot of the noise in the data. And so essentially what I did is a principal components analysis. And I'm not really a mathematician and I'm not really a statistician, but at a basic understanding, a PCA separates the signal in this square matrix into different orthogonal components, and you can then look at the amount of variability that each of those orthogonal components accounts for in your overall signal, and then you can separate the signal from the noise. So if you look at panel B, that's essentially what I did. Is I plotted the amount of variability accounted for by the component number, and it generates this nice sort of like elbow graph that looks a little bit like a scree function. And then you just got to find the elbow in that. So anything above the elbow you keep, anything below the elbow you toss out. So now then what you can do is for each of the participants in the experiment, there were 90 total. You have a weighting for each of the components, so you could figure out how much each participant in the experiment, how much their signal contributed to a given component in your PCA, and that gives you the matrix that you see in panel C. So this is like an 11 by 90 matrix because there are 11 components and 90 subjects. Then you could do that same basic multiple regression that I was describing in slide 23, but instead of using 5,000 or 55,000, 55,000 connections, I'm just using these 11 components. And so then I can use these 11 components in panel C to predict the symptom, let's just call it depression, in panel D. And then what I get from that is a coefficient vector in F. Now the vector in F, what that represents is how strongly each component predicts symptoms across subjects. All right, so almost there. Then what we can do is we can also use the PCA loadings, which basically tell us how much each individual connection contributes to each of these 11 components, and we can multiply that by this coefficient vector using matrix multiplication, and that gives us an output vector. And this output vector is 1 by 55,000. So essentially what this output vector now represents is the degree to which that specific connection contributes to the expression of depression across the 11 components. So we're sort of capitalizing on the signal and tossing out the noise. Does this make sense?
Yep, it makes sense to me, but please let me know, everyone, if you have questions, and we'll address them, or Nicholas will address them.
Okay, I promise this is the most complicated slide of the presentation. So if you're all on board with this, the rest of this talk is going to be smooth sailing. And I'm not sure how much time I'm taking, but I'll try to go a little bit faster as well. So if we jump now to slide 27. So all of this approach that I was describing is the approach that you would take for a single site and a single location, and essentially you're solving that MEX plus B equation for a single site and a single location, or a single site and a single orientation, and you get a single value for that. And that single value is the predicted change in symptoms based on how you think that stimulation at that site and that orientation is going to impact symptoms. Now, the really cool thing is that you can do this a bunch of times, especially if you have access to like a cluster computing environment. So you can iterate this approach across some sort of a predetermined parameter space. So in this model, essentially what I did is I computed this several times for various locations across an axis along the dorsolateral prefrontal cortex, going from posterior to anterior. That's plotted in the x-axis of the colorful panel in slide 27. And then for each of those locations, I also varied the orientation of the coil and iterated that across 24 equally spaced yaw orientations. So it's kind of like if you place the coil flat on somebody's scalp and then just sort of like twisted it along that angle. That's essentially what I'm doing. And so what that gives me is for each point in this I think it's an 11 by 24 matrix, you get a value that represents the predicted change in symptoms given that site and orientation of stimulation. And so this is where it gets fun. You can solve this equation. So once you have the loadings across subjects, you can solve this equation for individual subjects. So this is what I did in slide 28. So what you can see again is that same site by orientation matrix, where the cool colors represent a decrease in depression symptoms. So we're using the MADRS and the HAM-D, which are both standard depression instruments. And the whitish or the light colored circle is essentially the center of that is the optimal site of stimulation, and sort of the diameter of the circle just represents the variability in that estimate that we obtained using bootstrapping. And so, for individual subjects, you can see the optimal site and orientation of stimulation. And what's really cool is that it's very close to the site that's associated with the strongest subgenual anterior cingulate cortex anti-correlation, which I found, using an independent analysis. So the idea then is that we're replicating this previous work showing that this subgenual ACC anti-correlation is important for reducing depression symptoms, but we're doing it using this like completely novel modeling approach. If we jump to slide 29, you can see this is significant at the group level. So we used permutation tests. So these are the t values anywhere where you see red dots, which is sort of only in the bottom right corner of the second panel, is where the permutation tests did not surpass statistical significance. So you can see again that given left DLPFC stimulation in our model, we would predict significant decrease in depression symptoms. And the text on these figures now is representing different targeting approaches using either the 5 centimeter approach or different Brodmann areas, or like the BA9, BA46, or the F3, which is sort of a EEG 1020 based targeting approach. So you can see it largely replicates the previous work suggesting that the optimal site of stimulation is going to be pretty close to BA9, and between BA9 and this 5 centimeter approach. So what's really cool is that this model predicts what we have observed in the literature, or I guess it postdicts. So it's consistent with what we've observed in the literature. But it's based on assumptions. So it's based on the assumption that the primary assumption that it's based on is that the change in connectivity scales with the electric field model. We're getting close to 1 hour. Do you want me to go through these last few slides, or should I skip to the last slide and show the summary?
I mean, I'm fine. It's more how much time you have. We have time, but if you
All
kind
right.
of, yeah.
It's like four more slides. I think we can handle it. Sorry. I'll stick around for questions. So if anybody has questions at the end, I'm happy to answer them. But if we jump to slide 30 now, This is another data set that one of my colleagues provided and allowed me to use. And so in slide 30, this is the data that you really need to test this model. And the idea is that you want to collect resting state data before and after some sort of TMS intervention, excuse me, to plot the changes in connectivity and to see whether or not the model can predict those changes, and really to see whether or not the changes in connectivity scale with the electric field that is induced in the brain. So again, what we're trying to do then is we're trying to predict, or so we're measuring resting state data before and after theta burst stimulation, which is a type of TMS that is known to induce plasticity in the cortex. So we get measures for resting state connectivity pre-theta burst stimulation. We get measures for post-theta burst stimulation, and then we can calculate the difference between the two. And then to validate the model, what I did was, again, I processed the E field the same way I did in the previous study, where I calculate the E field. So this is slide 31 now. Downsample it to the cortical atlas, and then I create this E field matrix, which you can see in the top right panel. And then for the figuring out like what's the parameter space, for the previous study, those were unstimulated subjects. These were just subjects that I had, or participants or patients that I had resting state data for. These patients and these participants, not only did I have the resting state data, but I actually had the physical location that was stimulated. So what I could do is for each individual subject, I could define the parameter space centered on their site of stimulation. So that's what you're seeing in the blue set of panels below, where this big lightning bolt to the left is their actual site of stimulation. And then I just calculate for a few sites posterior and a few sites anterior, and then I calculate for the different orientations at the stimulation site. And then I can plot that in the same style of matrix that I had before. Now, critically, the values in this matrix no longer are the predicted change in symptoms, but the percent of variability explained in the pre-post changes in resting state connectivity by the E-field model represented in the top right panel. So, how well does the model predict the changes in connectivity? And if the model's doing a good job, it should predict changes in connectivity best at the actual site of stimulation versus a site that is not stimulated. So you should see the percent variability explained drop off as you get further and further away from the stimulation site. So you jump to slide 32. Indeed, that's what we see. So if you just look at the x-axis, you can see at the stimulation site, we get the strongest percent variability explained right in the center of the graph, and it drops off as you go to the left or to the right, or if you go more posterior or you go more anterior. And you get stronger values, at this center orientation, and the orientation that is 180 degrees from that, which is expected based on the physical models as well. So you get a systematic variability in the amount of variance explained in pre-post changes in connectivity as a function of both the site and the orientation of the stimulation. If you jump to slide 33, this just shows the group results. Again, if you average across the subjects and do the permutation tests, you can see that on average we're predicting, on average we're really predicting about 1% of the variability, but we're doing it fairly reliably. So we get a medium effect size. So we get like a Cohen's d of like 0.6-ish. And you can see that the only place where it's significantly, where the model is significantly predicting the changes in connectivity is right at the site of stimulation, shown in the center point of the graph on the left. And so, this is preliminary data. This is only 14 subjects. The grant that is getting reviewed literally today by the advisory council would be to expand on this model, try 2 different types of theta burst stimulation, and test it in 100 subjects per group with 4 different doses. So that would be a really, really good test of the model. Here we got a pretty okay test of the model, and there's good preliminary data suggesting that the changes in connectivity scale with the electric field induced in the cortex. But give me a few more years, and I should have a much stronger data set to either confirm or reject that assumption. So that's really where I'm at. So if we jump to the last slide, this just shows all the folks that helped out with this research. And so I want to say thanks to all them, and thank you all for listening. Sorry, I went a little bit over on time. I tend to talk a lot, but I'm happy to stick around and answer some questions if you have them.
Thank you so much for guiding us through this, this approach, and explaining to us really well how you managed to improve this, for the future, this type of treatment for individuals. I think that's quite amazing, because a friend of mine, he's now actually director of a clinic in Portugal, and they've been using TMS quite a lot, and they show already that a lot of patients are treated quite successfully. But with this approach to individualize the treatment, I think that will be even better. And so I think this is such an important work, and congratulations and thank you so much for doing this.
Yeah, well, what I think is really exciting too, sorry to jump in, is everything I showed today was about depression, but my research is focused on anxiety, and we don't really have the precise targeting approach for anxiety. But what we do have is a lot of resting state data and a lot of symptom data. And what I hope is that with this model we can relate the symptom data to the connectivity data in such a way that we generate novel targets. And so we find new places in the brain to stimulate that might be important for anxiety. And I just started a collaboration with a group from the VA department, where they have 500 participants with PTSD that have resting state connectivity and PTSD symptom measures. And I just can't wait to dig into that data set and really see if there's any novel places that we can stimulate for PTSD. I think that might be really cool. But I agree. Like I think like figuring out how to make TMS better and more applicable to different disorders is a really exciting field, and I'm just so lucky to be a part of it.
Yeah, I agree. Oh, that's really exciting. I'm really looking forward to learn more about your future work. Do you think that addiction maybe one day would also be somewhat ameliorated with TMS? I'm not sure, but
Absolutely. And there's actually a work ongoing. There's actually a substantial amount of work ongoing right now. I would say the expert in this area is Dr. Colleen Hanlon. She's done a lot of work with studying addiction and studying how to use TMS to treat addiction. I think a lot of her work was instrumental in getting TMS approved for smoking cessation. And really, what her work suggests is that the optimal site to stimulate for addiction is not necessarily lateral prefrontal cortex, sort of over the temple of your head, but really like the frontal pole. So if you were to place the TMS coil directly over the person's forehead, that's the optimal site of stimulation for a lot of the addiction disorders. And she's got an active program of research ongoing, and a lot of her trainees are pursuing this research. I actually think there's some folks at NPU that or NYU that came from her lab that are doing, these addiction-based TMS studies. So yes, I think not only is the answer to your question, an enthusiastic yes, but I think that we're really on the cusp of having that as, a treatment that's going to become more widely available for folks with addiction. So
Yeah, that's amazing. What do you think about, this is still somebody has to come to a clinic that has these, I don't know how expensive these devices are. What do you think about these ones that are on the market for home use? All this electric stimulation headbands type of things. Do you think they do anything? Because I think a lot of people are starting to use them.
yeah, so if you jump back to slide 5, you those sort of like wearable at-home headband, like transcranial direct current stimulation type applications, those really fall in this sub-threshold category of neuromodulation. So they're not really strong enough to induce action potentials. I think that, and I don't know, I don't know the clinical evidence well enough. So my understanding of the literature is that if you do a carefully designed study with a specific behavior and really precise localization of the TDCS electrodes, you can show behavioral changes with like TDCS or TACS. However, I don't know that there have been a lot of studies showing any clinical usefulness of this. So I think that it's probably still too early to tell, but I think that the effects, such as they are going to be, the effect sizes are going to be fairly small with TDCS and TACS. So, yeah, I think your mileage may vary with that. I will say though that they're fairly safe. So you're not inducing enough current to trigger a seizure. Typically, to my knowledge, I don't know if there have been any seizures induced with TDCS, but I don't think that there have been. And I think as long as you properly ground the equipment or run it off a battery, and as long as you properly place the electrodes, the risk of unintentional electric shock is pretty low as well. So I mean, I think they're safe, but the effect size is probably fairly small.
Well, thank you for those answers. Yeah, if anyone has questions, please flash your microphone. Serena, Katie, Dr. Shah, go ahead.
So I'm really fascinated in this work. Couple questions. One thing that really seems important about your model is that it sets up a more perhaps more optimal coil design, or multiple coils, or in taking different regions and tuning the magnetic field to hit more selectively. Have you looked at that? And is there anything you can say?
So I have not looked at different coil designs or different using multiple coils. I haven't done any of that work myself. I do have I do have some I do have some coil files that represent different coils that I can run through to see how, say, like something like deep TMS might affect brain connectivity. I haven't done that yet. That is something I do want to check out and see exactly how that works. And again, this isn't my work, but I know that there are people working to design multi-channel coils in such a way that you could trigger, different channels in the coil sequentially to actually guide the electric field, either in, XY space or sort of like in like orientation space. I don't know. I think those are really experimental, and I don't know that there's any of those commercially available yet at this point. But I think probably in the next 5 years, we'll start to see some sort of multi-channel coils that you can use to really optimize and direct current in a very controlled way. And man, if I could get my hands on one of those, that I think it'd be fun to do some studies with those. So yeah.
Well, it's interesting because you've got you're solving Maxwell's over the grid, and you've got your regression to suggest where those with the shape of an optimal effect or an encouraging effect would go in. It would seem that numerically you could sort of design the desired field, and then that would become an outsourced engineering issue to have coils to give that field. So it's kind of fascinating.
Yeah, that's interesting. I think I'm sure there are people working on that. I'm sure there are people more qualified than me working on that. I will say, if you jump to slide 13, all the work that I showed today, I was solving the equations for a specific coil type, and then it's the same, figure 8 coil that I use. So if slide 13 in the figure in the bottom left in that animation, those
Mhm.
coil dipoles represent the windings in the coil that I used. So the E field created and the solving of the equations is definitely going to be dependent upon the actual coil that you use. So I'd have to resolve the equations for each new coil that I wanted to test it on. And there's a really good paper by ZD Deng, colleague of mine, who essentially solved Maxwell's equations for like 50 different coil types in just a not like a full head model, but like a circular head model, just to show like the
Mhm.
current distribution. And one of the cool things is that as you design coils for different purposes, you start to realize that there is a depth focality trade-off. So you can create these like really complex coils that can penetrate deeply into the brain, but as you do that, they'll start to uh, create like more electric field across a larger swath of the cortex. So there's this depth focality trade-off that is sort of like a fundamental limit of coil design. Um, but yeah, so I don't know where I was going with that, but it's really
Well,
interesting
yeah,
work from
no,
the
because one area like in antenna design and beam steering, there's an awful lot of work in getting,
yeah.
yeah, getting that shape just right. But then, so in terms of the connectivity, though, what's really fascinating, and I promised I'd get around to astrocytes. The one function they serve is to in a very non-local and spatial way connect different regions of neurons, and get them to fire in synchronicity. And I'm wondering if insights there could possibly improve the model, not just in terms of the connectivity or the correlation, but perhaps also the phase and the timing, because the support of the synchronicity comes from these waves at certain frequencies that resonate. And so I'm wondering if there's if that's a rich ground to improve the model.
I think that that's really interesting. I think sort of to broaden the question a little bit, like understanding the cellular mechanisms of TMS stimulation is really critical for understanding how they're going to affect the cortex and connectivity and excitability. And one of the really exciting developments in the field is the development of theta burst stimulation, which uses really rapid bursts, so 50 hertz bursts of single pulses that are then nested inside of a theta wave, so a 5 hertz frequency. And this is all based on electrophysiology work done either in hippocampal slices or in cells in the hippocampus, showing that these theta waves are really important for synaptic plasticity. So they developed theta burst stimulation as a way of inducing plastic changes in synaptic connections. Again, it's not about astrocytes, but inducing plastic changes in synaptic connections with many fewer pulses. And now, a typical TMS treatment using 10 hertz stimulation is going to last about 40 minutes, but you can get the same effect using intermittent theta burst stimulation, you doing a session that lasts only 3 minutes. And now there are groups that are starting to do what's called intensive theta burst stimulation, or I think the main model now is the Stanford, St. Stanford something applied intelligent neuromodulation treatment, something like that. But basically, they're doing connectivity targeted intermittent theta burst stimulation, but instead of doing one session a day, they're doing like 10 sessions a day, and they're cramming a whole course of treatment that would usually last 4 weeks into a single week, and showing just some really profound decreases in depression symptoms. And it's all based on this fundamental cellular neuroscience work showing that this particular wave of stimulation has these plasticity inducing effects. So to get back to your question, I don't know the state of the research looking at how TMS affects astrocytes and whether or not we can capitalize on specific frequencies to target astrocyte function. I think that there is some work looking at how to control the shape of the TMS pulse that might affect different cell types as well. But I think that those studies are probably ongoing. I just don't know them at this point. But I think that it's really exciting.
Well, yeah. So what's at least from some recent Nature reviews, what's part of the astrocyte calcium waves? It's in the theta region to low gamma, and they support the plasticity. I wouldn't be surprised if the astrocyte function is behind the what you just described. So it's really, there's a lot of just new stuff coming out on astrocytes, and it's really it's showing profound effects in anxiety and depression and dementia and in other higher cognition. So I wouldn't be surprised if it's, if they're finding benefits with theta waves, that there's some implication for astrocyte being a part of that cellular mechanism. But anyway, fascinating talk. Thank you. Thank you very much.
Thank you.
Yeah. Does anyone else have questions? Dr. Shah, go ahead.
Yes, thank you so much, Nicholas. That was a wonderful, research that you just shared with us. My question is about, chronotype. We know that we have a different population and they have a different circadian preference. Also, we know when we are talking about the chronotype, people can choose to be in what, level. Also, there is a relationship between chronotype and the genetic performances. Somehow, I just look at the slide that you just provided, and some of the areas that you reported in your study was pretty much matched with the sleep deprivation and problem that we have with some of the patients that we can use the TMS. I was just wondering, can we have more information around that? If you have more information, that would be nice. Thank you.
Yeah, I will say I'm not an expert in chronotypes or insomnia or sleep deprivation. I do know that sleep disturbances have a profound impact on both depression and anxiety. In fact, I'm part of a project that's starting to look at some of the relationships between insomnia and the development of anxiety and depression. Um, and I have no doubt that, there are mutual relationships between anxiety, depression, insomnia, sort of influencing one another back and forth. And so the treatments for depression, whether they are directly targeting depression and minimizing, sleep disturbance systems just by symptoms, just by sort of reducing the overall burden of psychopathology on the subject, or whether or not they're targeting specific insomnia locations that share overlap with depression. I'm not sure the answer to that. I do know that there are a few active studies going on looking at using TMS to treat insomnia, or to study and understand insomnia. I think that there are studies outside of the prefrontal cortex. I think there are some studies looking at parietal stimulation. Again, I'm not an expert in that field, but I have no doubt that the circuits would be overlapping just because the symptoms are overlapping as well. So um, ideally, if you could you know maybe treat the insomnia, maybe you could reduce a lot of the depression and anxiety symptoms as well. But I don't know. It's not I'm not an expert there, so take my answer with a grain of salt.
Sure, thank you. And just out of curiosity, because you had 90, participants, right? So what's the average age they were?
Ooh, that's a good question. I could look it up in the paper. I haven't looked at those data in a while. I want to say they're slightly older than the average college age population for like healthy volunteer studies. I want to say average age is probably in the 30s somewhere. Um, yeah, those are definitely some of the details that I haven't committed to memory yet.
So the first time, do you remember how many they were, and then you ended up to 90? For example, your main sample was 100 people, and then you reduced to 90, or 90 from the beginning to the end?
So I know that we had more than 90. We had to remove several people. A lot of it was just due to either movement during the MRI scans or artifacts in the MRI scans, or just sort of incomplete data. I'm just looking at, yeah, I think the average age was somewhere around like 28-ish, plus or minus 8. And then I think we had around 108. Oh yeah, we had 118 participants we had a cohort of like 25 or so healthy participants. I'm looking at the paper now. Yeah, I know we did have to throw some people out. I can't remember exactly how many we threw out, and for what reasons, but it's definitely reported in the paper somewhere.
So thank you, Nicola.
Sure.
I saw Deep, you had a question. Go
Yeah,
ahead.
thank you, Katerina. Yeah, thank you, Nicholas. I have a simple layman question. What is the sort of neuroplasticity and and the treatment, like how much they endure, what kind of studies, what's the longest studies that have been done? I imagine what you're doing is fairly recent, so you probably need more time, but generally in the area, uh, what is going on with, in terms of the treatment enduring and the actually the plastic, the neuroplasticity.
Yeah, so a typical course of treatment for depression, like the FDA approved treatment, you would come to the clinic for like 5 days a week for about 4 weeks. And on each of those days, they would deliver 40 minutes of 10 hertz stimulation. Um, that's like a typical. That's like the typical treatment for depression. The the theta burst treatment for depression, which was more recently approved, it mirrors that very similarly, where you would have like the person would come in 5 days a week. They would come in for about 4 weeks, but instead of having a 40-minute session, they would receive only about 3.5 minutes of stimulation. The more recent the more recent studies are using accelerated or intensive TMS, where they group more sessions in a day, and they have longer days but like shorter courses. So I mentioned the Stanford group are delivering 10 doses per day of theta burst for a full week. We have some trials ongoing in our center that are using that approach. I have a similar approach that I'm starting with anxiety, where we're doing 8 sessions of 1 hertz stimulation a day for a week. As for like the neuroplasticity and the neural changes, I think that's really hard to quantify. So a lot of these studies are done retrospectively, where they'll collect resting state data before the TMS, but not necessarily after. A lot of the studies that look at before and after changes will often look at within session changes. So you record resting state, stimulate, and in the same day, you record resting state again. And there are much fewer studies that say give a course of stimulation and then measure the resting state before and after. And I think primarily because it's a fairly expensive study to do. The studies that have found it or have done that have really focused on specific specific seed-based connectivity, where you just take one region of the brain and correlate it with a few other regions of the brain, mostly to avoid issues with like multiple comparisons. A lot of what's been found with pre-post changes in connectivity have shown really fairly modest changes in downstream connections from the site of stimulation. But I think a lot of those studies really suffer from the fact that they're using a seed-based approach as opposed to sort of an e-field weighted approach, which I'm using in my current work. And they're not really looking at whole brain changes; they're just looking at single seed-based changes. So I think that we have a lot of room to grow and a lot more to learn about the plastic changes that occur following um, neuromodulation. And I should also say, different durations and different patterns and different types of stimulation are all going to have differing effects. And really, the parameter space to explore is fairly large. So I think there's just a lot of work to be done. I guess is the bottom line.
I bet, I bet. But what is what have been the longest sort of studies in terms of that somebody got treatment and then they came back maybe 2 years later or something like that,
Okay.
and then they were looked at again? And just another small question: any side effects? Those are my questions.
So the first question, I think the longest pre to post measures of connectivity that I know of are on the order of like a week to a couple of weeks. I think it's definitely more common to measure symptoms for several months to years after TMS as opposed to connectivity. And I will say, while the changes in symptoms do last, oftentimes, in a therapeutic setting, individual patients will need sort of like top-up sessions, or they'll need to come back to receive sort of maintenance sessions following a course of TMS, in order to for the effects to last a little bit longer. Now, side effects. There are really two categories of side effects. The first one is really a risk factor, and that's the risk of inducing a seizure. So while TMS is generally thought to be uh, below the threshold for inducing seizures, there have been seizures induced using TMS. So in a typical TMS laboratory, we do a lot of things to try to mitigate that risk. We they don't have any risk factors for epilepsy. We screen them to make sure they're not taking any seizure, any medicines that lower the seizure threshold. And then we do another technique known as motor threshold testing, um, where we calibrate the intensity of the stimulation to the individual's brain. So what we do is we basically find their motor cortex, the spot that makes their thumb twitch, and we dial the intensity of the stimulation down so that their thumb twitches about half the time, and that's the threshold between being able to activate the brain cells and not being able to activate the brain cells. And then we use that as a basis for how to define the stimulation during their course of treatment. And then the other sort of category of like side effects are more commonly experienced, and the main one is like headache or discomfort. So the TMS coil induces a magnetic field, and that penetrates through the skull and that activates the brain cells. But it's sort of going through everything in between there, and the stuff in your periphery runs on the same electrochemical processes. So your nerves and your muscles and everything. So depending on where you hold the TMS coil, you'll have different peripheral effects. So let's say I place the TMS coil over the back of your brain, say on your parietal cortex or your motor cortex. You don't really have a lot going on there in your periphery. It's just like your skin and your skull. So what you'll experience in that case is sort of like a tap or a mild shock or a pinprick or a buzz. And it's really just the magnetic field activating the skin cells in that particular location. But as you move the TMS coil towards the front of the brain, you you start to activate the nerves that control the face and the muscles that control the face. So the primary thing that you feel is like your forehead starts to clench, or your jaw might clench, or you might feel like a weird sensation in your jaw, or weird sensation in your teeth, or you might blink, or your eyes might water. Those are all just sort of natural reactions to the TMS pulse activating your peripheral innervation of the face. And depending on the intensity of the stimulation, some people can find that unpleasant and it can induce a headache. But generally, headaches have been fairly mild and they tend to go away with like over the counter medication. Um, yeah. And so I would say probably, depending on the study, maybe like 1 in 5 people end up having a mild headache from the treatment.
I'll ask a question if we're done with that line. I was curious if there is any applicability to this in a chronic SARS-2 case, if you're aware of anything like that, or if you see any sort of applicability for that condition.
Could you repeat the condition?
SARS-2, COVID?
Oh, COVID. Yeah, I don't know if there's any work being done on that. If there is, I'm definitely not aware of it. I don't. Um, yeah, I'm not too sure even what the target would be for that. Maybe there are some symptoms of long COVID, like and I'm not an expert in COVID, like lethargy or sort of lack of energy or something like that, that maybe could be treated with central stimulation. But I don't know that it's really the right technology for treating COVID in any sort of way.
The reason I asked was some patients are being diagnosed with anxiety, depression, and things of that nature, but obviously it could be due to different mechanisms.
Yeah, and I think whatever the mechanisms, I think that if somebody has if somebody has failed other medication treatments for their depression or anxiety symptoms, then I think they might be a good candidate for TMS, regardless of sort of what the instigating factor for their depression and anxiety symptoms. But I don't know that I've seen any applications of it specifically for depression and anxiety symptoms in COVID patients.
Thank you so much for your answers.
Sure.
Hello, Dr. Nicholas. Thank you very much. I'm sorry if I took someone's turn, but I just had a question. Um, Especially while working with the depression patient with depression, as you know, like there is a very famous side effect of depression, like withdrawal, or maybe the patient with depression will have like high expectations about this treatment. And as I know, I hope I'm right, like this usually it will be a prolonged procedure requiring more sessions. So have you maybe experienced like some of the cases they will have more maybe let's say disappointment while they don't have patience to examine the I mean the results from this treatment, while and also about their commitment to treatment. They are coming every session, or do like have you faced situations where the patients just might maybe stop coming, or maybe they're expecting everything, all their problems to be solved, giving their mental health is fragile at this moment and at this time, I mean. And another question, what about patients with the migraine? uh, if patients are known to have migraine for a long time, or are they recommended for this type of treatment? And my last question, I know so much, I'm sorry. My last question, is it like does it stimulate the transmitter to produce more dopamine? Like if the patient was, for example, misdiagnosed for depression and he received this type of treatment, would that make anything worse or nothing will happen? Thank you. I'm sorry for so much questions.
No, that's okay. Thank you for the questions. I will say it's getting a little bit out of my expertise. I don't, in terms of the outcomes for depressed patients, I don't know that there are a lot of depressed patients that are withdrawing the study from the studies just because they don't have the anticipated effects. I will also say a lot of my work is in healthy volunteers, or a lot of my sort of like hands-on participant contact has been in healthy volunteer studies. So I don't know that I necessarily have the clinical background to really give you a good, satisfactory answer to that question. I will say across the center, dropout for our studies has been fairly low. But we also provide compensation for our participants, so that may be a mitigating factor. The second question, I forget. The third question was whether or not the TMS induces changes in dopamine receptors, and I think that there is maybe some preliminary evidence that there might be some changes in dopamine receptors at the site of stimulation. I think that's I think there needs to be more work in that field, and so studies looking at dopamine receptors using PET. I'm not really a PET expert, but positron emission tomography before and after TMS, I think would be a really good avenue to sort of test that hypothesis. I have a friend who does alcohol use disorder work, and she does some PET imaging in that population, and we've talked about trying to do some kind of a project in alcohol use disorder patients, looking at dopamine receptor expression following TMS, but we haven't really gotten anything off the ground at this point. And the second question, I do not remember.
It was about a patient with chronic migraine. Are they?
Oh, yeah. So there are TMS treatments for migraines. Actually, I think one of the hypotheses about migraines is that they may be related to hyperexcitability in the cortex. So I think that there are some treatments, TMS treatments, aimed at reducing cortical excitability that have been shown to reduce the frequency of migraines. I don't know that it's FDA approved at this point, but I know that there's research going on in that direction.
So I mean, doctor, this the migraine is related to being hyperactive, or did I get this wrong?
So I think that that's the hypothesis that that is driving the TMS research into migraine treatments, is that one particular mechanism behind migraines may be hyper excitability in the cortex. So if you can design a TMS treatment that reduces cortical excitability, you may be able to reduce migraine frequency. But again, that's definitely not my field, but I think that there are studies ongoing with promising preliminary data in that direction.
That's very exciting. Actually, I am diagnosed with ADHD and I have a chronic migraine. I never linked both of them together. I'm gonna look into that. Thank you very much, doctor.
That's interesting. I've never made that connection either, so there might be something there.
Thank you very much, dear.
Yep.
Yeah, thank you so much for answering all these questions. I know we went way over the time we planned for it, but we
It's
really
okay. I haven't finished my beer yet, and there are no screaming kids, so I have
Oh,
time.
okay. Oh, you still have time. Okay, perfect. I wanted to make sure to check in. And yeah, if anyone has more questions, Jamie, Kyoko, Victoria, you didn't have a chance to ask, or anyone in the audience. Eric is here, Dr. Olo is here, and many other of our friends. So ask questions. We still have a little bit of time.
I was just trying to reconcile. I thought you said that you mostly had healthy volunteers, and then but you did studies on depression. I was just wondering, but maybe I didn't get that.
Yeah, so the data that I presented today are from two studies that were collected in our center, but I wasn't necessarily a part of data collection. So the first study was an ongoing study where they just measured resting state connectivity and symptoms in a large cohort of individuals with depression and anxiety. So that was collected mostly before I got to the center, and I was able to just take the data and analyze it. The second study was an ongoing depression treatment trial led by Dr. Desmond Othis, who's co-director of the center. So I was able to again analyze his data, but again, I wasn't really involved in the collection of those data. I've done some neuroimaging studies in individuals with anxiety disorders when I was at the NIMH. And I have a grant that I submitted, actually, a grant that I will submit in about 2 weeks or so, that where I'm proposing to do a TMS trial in GAD patients. But yeah, my background is in experimental psychology, and I really came to a psychiatry department so that I could partner with these clinicians and bring a lot of like technical skills and rely on their clinical expertise and access to patients to do some of these more clinical trial oriented studies. So it's definitely a team science effort.
Yeah. Does anyone else have a question? No, not. Yeah, I wanted to ask if I saw some years ago there was an MIT study that that changed with TMS kind of a little bit the ethical evaluation of some like ethical questions, like scenarios. Do you see any changes in like character traits with TMS? I know it's a more general and probably it gets a little bit away, but it's something that made me curious. But then I didn't see too much of a follow-up question in that way.
Yeah, I will say in my studies, I do not, and I think in a lot of the clinical studies, I don't know that they see a lot of those character traits, changes in character traits, and I think there are a couple of reasons behind that, and this is me speculating at this point. I think um, I think for a treatment to be effective, you kind of have to be open to it being effective and open to like the changes that the treatment is going to have. And I think that it works for a lot of these clinical disorders, specifically for that reason. You have people that are motivated to get better in these treatment trials. For these more experimental, like cognitive and behavioral and social neuroscience studies, I think that the effects that you get are going to be much more subtle, and they're probably not going to be very long lasting. And I think as with any sort of like personality trait, um, I guess what I want to say is like any sort of personality trait is going to be also much more difficult to move as opposed to sort of like mood and anxiety symptoms, just because I think they're fairly ingrained into our sense of self. But again, though that's a purely speculative answer.
Yeah, thank you. Thank you for that. And Dr. Olu, you joined the stage. Did you have a question?
Yeah, and I apologize for coming late. I just had a couple questions. Nicholas, the paper cited in your slides from 2021, I was wondering what journal that was published in?
I think you're referring to this proof of concept study. That was neuropsychopharmacology.
Oh,
I
okay.
think the link to it should be in the slide notes.
Okay. Okay. Great. Because I definitely wanted to check that out in more detail since I came late. I was also wondering, it looks like your networks were based on resting state fMRI, and I was wondering if you've also looked at looking at structural connectivity from diffusion weighted imaging to look at the influence of that or include that in your method.
I haven't personally looked at that. I am part of a study that, again, is led by one of my colleagues, Desmond Othis, that should come out soon. I think in like Science Communication, where they looked at structural connectivity between the DLPFC and the amygdala, and how much that played a role in symptom change. I forget exactly what the findings were of that, but it was a pretty interesting study, pretty interesting analysis of the data. The other thing that I've done in some of my anxiety focused work is to use functional activation, like brain activation during a working memory task to guide targeting. So let's say I have a hypothesis about how working memory and anxiety interact. So I have the person do like a working memory task, identify the DLPFC cluster most activated for the working memory task, and then stimulate that and measure the effect of that on anxiety. I've done that across a couple of studies and found, direct links between stimulating DLPFC regions associated with working memory and changes in anxiety after that. The story is a little bit unclear, but there's definitely some sort of a connection there. I can, if you email me, I can send you the links to those as well.
Yeah, I'll definitely follow up. It's really interesting. I've seen a increasing number of studies where they're using that similar type of paradigm, but instead of a working memory task, it might be some kind of task actually involved in the therapy, like a cognitive restructuring type of task,
Yep,
and then looking at the areas that are activated for targeting for TMS to enhance that ability to do that
yep,
skill that they're trying to learn as part of their therapy to kind of accelerate the therapeutic response to the therapy.
absolutely. And then I think to push it one step further, the you may be able to um, further increase the effects of the TMS if you control the temporal context during which the stimulation occurs. So let's say if you're stimulating a working memory region, maybe stimulate that region while they're doing the task, so you can sort of take advantage of the co-occurrence of the DLPFC activation and the DLPFC stimulation to facilitate some sort of synaptic plasticity effect. I think that's definitely an ongoing field of research, and one that I know NIH is really interested in like pursuing.
Yeah, absolutely, and it makes a lot of sense as the time that it takes for stimulation gets shorter and shorter, like for intermittent theta burst stimulation where you can get it down to 3 minutes for every treatment session. You have to think about bringing a patient in, what are they going to be doing with the rest of that time, and so having them
Yep,
engaged in actual therapy as part
Yep,
of receiving the TMS makes so much sense from a pragmatic standpoint as well as a clinical one.
absolutely.
Thank you. And yeah, I'll definitely email you and follow up on getting those references.
Uh, I had another question that popped up, and you considering you're a psychologist, and kind of related to what Katerina was talking about, because I'm interested in the behavioral aspect of this. And so you had remarked that that some of the traits that are, I guess, deeper parts of our personality would not be affected. But are there, I mean, psychological disturbances which could be more innate to us, or maybe genetic in that way, that would be tied in? I mean, that would be also kind of innate, that are not maybe just products of later childhood development or something like that. Like that's what I'm trying to get at. That you see what is my question clear?
I think so. The the only thing that I have that I could think of to say in response to that is a lot of my work on anxiety is focused on trying to stimulate and potentially inhibit the regions of the brain that are important for fear and anxiety expression. And we know that those regions, developed very early in our, I guess, like phylogeny, and they're very preserved across species, and they're very they're very difficult to change and to target. And so a lot of my work is to try to figure out how to either directly inhibit those regions through sort of inhibitory stimulation, or inhibit them by boosting the top-down inhibition circuits that we think are important for regulating those behaviors. I would say that I've been met with mixed success. I think that I've found some really interesting findings, but there's still a lot of work to do. But I think the closer a trait is to your fundamental, feeling of self, like the harder it is going to be to augment. But again, that last sentence is pure speculation.
So are those the areas that you're inhibiting? Would you consider that they also have some useful functions that could be inhibited? Um,
Yeah,
yeah.
absolutely.
And what
Yeah,
could
and
some of those functions maybe? If you want to give an
you know, I,
example.
I I think so. So one of the studies that I'm doing, or one of the series of studies that I'm doing, is looking at parietal cortex inhibition for the reduction of anxiety. And we know that the parietal cortex is a region that's really important for attentional orienting and recognizing stimuli for the environment. But overactivation of this region could contribute to the hypervigilance that you see in something like PTSD. So I think that while there is a danger of pushing the circuit too far in the direction of sort of maladaptive adaptations, I think that that risk maybe is outweighed by the potential benefit of reducing somebody's, say, like hypervigilance symptoms, and somebody who's experiencing severe PTSD. I don't know. I think it's a balance, and like I said, I'm not a clinician, but I think those types of decisions should be made on a one-on-one basis, in a sort of clinician-patient relationship. And those sort of risks and benefits need to be weighed in that sort of a context.
Yeah, thank you. I get that. Yes, thanks.
I have maybe a last question. I don't know how much time we have. So, how do the because you can apply also different frequencies, right? To either have a more excitatory effect or a more inhibitory effect. So, how does the frequency and the timing of the stimulation go into this personalized, basically TMS approach? Is there like a general, for this disorder, we just use this type of stimulation, or would you go by I don't know, brain imaging or the symptoms? Like, does every symptom have basically a set frequency that you would apply, or would that also need a more personalized approach, basically?
Yeah, wow. That is such a minefield of a question. It's really like that is like the core of the whole TMS field. And I think that it's really the most interesting and difficult question that we're sort of faced with right now. And it's also the question that it's like almost like the hardest to get data on. So I'll tell you like how it works now. So much of the data that we've collected to understand how frequency or pattern affects excitability has been collected primarily in studies looking at motor cortex excitability. And so the way that they do that is they'll stimulate, say for 600 pulses with continuous theta burst or intermittent theta burst, and then they'll measure people's thumb twitches before and after that. So if you send a single TMS pulse, you can make the thumb twitch, and then you can measure how hard it twitches with like EMG electrodes, electromyography electrodes. And so you can say like, oh, okay, so I give 600 pulses of continuous theta burst stimulation, and then I measure the EMG response before and after, and I see that their EMG response went down. So continuous theta burst decreases excitability. But then, you got to factor in the time between sort of the intervention and the measure, whether or not like the TMS effects are acutely on board. So whether it's like within session versus say something like 24 hours between session. And then, you have to make the assumption that the effects that you observe in motor cortex are going to generalize to completely different areas of the brain, like, say, like the dorsolateral prefrontal cortex. But it's harder to collect the data to directly confirm or refute that hypothesis just because we don't have any really good behaviors that you can measure immediately after stimulating the DLPFC to track that change in cortical excitability. So it's a really, really difficult question. The project that I'm proposing that got reviewed today, I will basically measure changes in functional connectivity before and after, or change in connectivity after several doses of either continuous or intermittent theta burst stimulation. So I won't really get any data on excitability, but I will get data on changes in functional connectivity, and I could use that to scale the E-field model effects in the model that I'm working on. But I think it'll be just one piece of the puzzle. In terms of like what's being done now, typically for a given disorder, there'll be a site and a pattern of stimulation that seems to show an effect. But to get to that point, a lot of times it's a little bit of a trial and error. So yeah, I think we're still really working on figuring out the best ways to understand how TMS changes excitability and synaptic plasticity outside of the motor cortex. And I don't think we're there yet, but that's a good place to be in science because you at least you got a job for a few years. So
Exactly. Is there a way to have people talk, do something like, would that give more insight? Like when you do brain surgery and people have to answer questions to know that you're not hitting something wrong.
yeah.
Would like a way to ask them questions related, let's say depression, and we know that, I don't know, they would answer something in a kind of expectation that everything will go wrong anyways or something like that. But would there be some immediate effect that you would be able to see a change right away, and how they would answer the same question again during the stimulation, and that's when you hit the right frequency? Or is that complete nonsense what I'm asking?
No, I mean, I think that is the holy grail of clinical TMS neuroscience. I don't know that we have anything like that at this point. I think some good candidates for that might be something like I'm working on using potentiated startle as an index of anxiety, although that kind of has its problems with TMS that maybe I'll save for another talk. Or perhaps something in the way of like eye tracking or pupil dilation or something like that, or maybe some sort of a reward task might be sensitive enough to do that. At this point, we don't really have anything that's really acutely sensitive to DLPFC stimulation that we could use as a biomarker. But if we got that, that'd be really great.
What about minor pain? Isn't there a difference? I don't know. That's the problem, right? Probably it's not the same thing for everyone, but isn't there a difference in pain perception too? And depression?
Um, yeah, maybe. I guess you could maybe deliver a painful shock sort of before and after the stimulation, something like
Yeah,
that.
like
Um,
finger, like a tiny, like this prick test that you'd sometimes do with mice too.
yeah,
Tiny ones, not something that really hurts, but maybe it would be different, perceived like more severe. I don't know. I don't
y-
know, right?
yeah. That's possible. I don't know.
Or it tastes good or not. I don't know.
Yeah,
Interesting. Well, it would be interesting. It's a interesting problem.
yeah,
But
definitely. The other thing that complicates that is one last thing, is a lot of the off-target effects of the TMS can have impact on your acute mood and your sort of acute feelings. Like, so, if you're getting, theta burst stimulation to the prefrontal cortex, it's, you're having your forehead constantly jerking and it's sort of like an intense, sometimes painful feeling that may also change these same variables that we want to use as indices, complicating that. And we have sham stimulation, but it's not quite good enough to um, use for that situation. But yeah, I don't know, that was getting a little bit rambly, but yeah.
yeah, thank you so much. It's getting kind of late. Does anyone have maybe a last question, or we all good? Yeah. Okay. Thank you so much for taking the time again. It's fascinating work. Now I'm all
Sure, no,
curious.
this is great. This is good fun.
I'm even more curious now. And I wish you all the best and a lot of luck and grants and patience that go along and
Yeah.
everything works
All right.
out.
Thank you very much. I'll be sure to tune into some of these other talks. Like I said, I think this is a really cool group. So
Yeah, thank you. Yeah, that would be awesome. That would be amazing. Come back anytime and give us updates anytime you would like. And yeah, please come back. That would be wonderful. And participate in our discussions.
that sounds good.
Yeah,
Everybody, take care.
take care. Thank you for taking the time. It was great.
Thank you so much.
Thank you for your
Okay, bye
time,
bye.
Doctor.
You're welcome.
And for everyone in the audience, thank you so much for asking amazing questions and being here, participate or just listening. Follow the club if you like rooms like this, and we'll have our next room on Thursday, 1 p.m. EST. Dr. Kachia Paglia will talk about massive gravitons and if they are good dark matter candidates. And then in the evening on Thursday at 9 p.m. EST, we will have Dr. Moses talking about the BCI for decoding speech in paralyzed person. And he has actually unpublished updates that he will share the first time with us. And then on Friday, we'll have at 2 p.m. EST Dr. Lepora talking about artificial 3D printed robot skin that has that feels like a human, which is also really interesting. And so yeah, come back and thank you so much, everyone. Again, enjoy the rest of your evening or morning or middle of the night, like Jamie. Go to sleep. And thank you so much, everyone. Okay, I'll close the room in 3, 2, 1. Bye, everyone.